the opportunity to share with you my thinking process and how I got involved in looking at plants, because I'm not a botanist. I'm a biological anthropologist, a human biologist. But how I kind of arrived at this thinking is, for me, it's rewarding to share that with you, because all science is integrated. And we tend to forget that as we partition our lives and partition our knowledge into little dysfunctional segments. And for me, this experience, this journey, looking at plants and genes and evolution, has made me think in much richer, more integrated ways. So let me go ahead and get started. Okay, so I'm going to concentrate on phytochemicals. And these are just chemicals in the plant. Plants are incredibly sophisticated. I have such respect for plants because we imagine we are the lords of the universe kind of thing. But really, in terms of chemical evolution, plants are so much more sophisticated. And they have had more time to evolve a wide range of defenses, compounds that attract us to them, compounds that repel us. They're really running the show, but we like to think that we are. So I had been working on this book called How Plants Domesticated People. And I still haven't managed to get it finished, but it's something that I'm working on, because every day I'm learning more about this complexity of human-plant relationships. And I hope to share a little bit of that with you. Now, these are some of the phytochemicals, or the chemical compounds naturally found in plants that we eat every day. And what we've discovered over the last 20 years has been that it matters what you eat, and your exposure has an impact on your biology and your behavior in unanticipated ways. We didn't have to wait for the Twinkie defense, for those of you who remember the Twinkie defense out in San Francisco as an excuse for murder, to realize that what we eat does influence, if not our genes directly, then the epigenome, that is the chemicals that surround the genes that will influence how genes express themselves. So usually when we talk about phytochemicals, we're talking about wild foods, because wild foods are so exotic and they're often, we think of them as untampered, and that they have their original phytochemical contents and so forth. But even in the domesticated foods, the foods that we have cultivated over the last 10, 12,000 years, they have still, many of them still hold on to a lot of these poisons, these phytochemicals, which in large doses can be poisonous, but phytochemicals that that mimic some of our own metabolic compounds. And that's what makes them so fascinating to me, is that this division between plants and animals is getting fuzzier by the minute. Plant domestication is something we're still trying to understand. The old theories were that all plants were domesticated in one place, and then there was this diffusion to other places in the world. But it seems to be something deeply embedded in our own genomes, this point that comes in human history, where different populations independently develop these close relationships with the plants that are around them. I don't fully understand what would make someone who was a gatherer and a hunter start paying attention to that plant. And you think of what our ancestors must have gone through, 15,000 years ago, armed with what we would call very primitive weapons and very serious competing carnivores and some hyper-carnivores. So plants must have been a reliable alternative. Something you could count on, if you could mentally map where the plants were located and when you're supposed to eat them, because plants have their rhythm. So it means that the human population, our early ancestors, had to tap into the rhythms of the plants. And some primatologists have hypothesized that ability fostered increased neural networks, more complicated mental development, because we had to remember where in the forest or where in the savanna a particular plant would be, when it was ready, ripe to eat, what were the signs that it could be eaten, what other signs meant don't eat it yet, So we have all these different locations in the world where modern humans now have left Africa, they've established themselves in places other than just Africa, and we see domestication in places like New Guinea and in what is now China and in the Americas. And it's all very fascinating because it wasn't like there was a rule book that said, now this is how you domesticate. It seems to have emerged with the populations. Maybe it's a part of our nature. Our ancestors were just as bright as we think we are, maybe brighter. We were talking about it, maybe they actually had something on us. They had fewer distractions. But I'm sure that they recognized that certain consumption of certain kinds of foods elicited certain physiological responses in them or certain behavioral responses in them. And so those wise ancestors, I'm sure, began to see correlations. Even if they didn't understand the level of causation that we focus on now, they would have seen correlations between what they ate and how they felt, what they ate and how other organisms responded to them. And that sets the template for talking about human-plant coevolution and ultimately human-plant parasite coevolution. So the story I want to tell you starts for me in West Africa, in a small country called Liberia. My husband's a nutritional scientist, and we went to Liberia to do our dissertation research back in the late 70s. And we got to Liberia, and it's really only about 200 miles across from Robertsport all the way down to Harper, with Sierra Leone, Guinea, and Cote d'Ivoire bordering Liberia. And Liberia was a very interesting place because even though this is a very small country, we found that the frequency of sickle cell was significantly different from one part of the country to the next. Yet malaria was endemic throughout the country. So I'm scratching my head and thinking, I had come there to do this project on genetic adaptations to malaria. But in this tiny country, there were significant differences in the primary genetic trait that we thought at the time controlled response to malaria, to falciparum malaria. And I began looking at that, going back to the literature, and at that time, colleagues at Rockefeller and Albert Einstein were looking at cyanate salts as, with their urea base, as a way to inhibit sickling. And I thought, wow, that's really interesting. I wonder where I could get 0.3 molar cyanate. And then I realized from plants, from cassava. So in this pattern in Liberia, what we saw was that in the southeastern part of the country, down here near Cote d'Ivoire, very low frequencies of sickle cell. But in the northwestern part of the country, high frequencies of sickle cell. Remember, I said this is only 200 miles across here, and probably about 50 miles, 75 miles deep. So very small country, not a lot of cultural diversity, and yet significant differences in the frequency of sickle cell. And then I realized that in the northwestern part of the country, people were eating cassava, but only as a green vegetable accompaniment to rice. And in the southeastern part, they were eating cassava every day. It was a major staple. So their exposure to the plant phytochemicals was much greater in the southeastern part of the country than in the northwestern part. And so I kept thinking about this. Well, so how would this influence the frequency of sickle cell? And how might the diet kind of knock the legs out from under the frequency of a disorder like sickle cell? Because as we know in genetics, there's no free lunch. You know, you get a gene that looks adaptive in one setting, but it usually also has some deleterious aspects. What the researchers at Albert Einstein and Rockefeller had found out was that if you exposed red blood cells to cyanide, they were giving it orally, or cyanate salts, or urea-based compounds, that you would get something called carbamylation. And what this did was it simply modified the protein such that the red blood cell had a longer lifespan, it held onto the oxygen better, and it was more resilient against deformation as it passed through the capillaries. So this was all very good for people who had sickle cell anemia. It meant that their red blood cells would behave more like normal red blood cells. Of course, the downside was that the metabolism of the red blood cell is very similar to the metabolism of the lens in the eye. So these people were becoming blind as they were also being treated for sickle cell anemia. And I thought about this and thought, well, how do we establish that balance between poison and non-poison? How do you manipulate the diet so that you get enough of the toxin to interfere with an unfortunate and deleterious metabolic event, but not enough poison to kill the person or produce a whole nother set of problems? So here's the cassava root. It's tapioca. If you say tapioca, you would hardly recognize that this was the root that it came from. It's a tuberous crop. It's pan-tropical, which means that it's grown throughout the tropics. And when I came back from doing my dissertation work and I started working at Berkeley, and at Berkeley, as you probably know, they always want to know, what have you published? Where is it? Give it to me. Show it to me. So this whole publish or perish business. So I figured if I worked on cassava, I couldn't lose because it's full of cyanide. So I'll either prove it's good for you or I'll prove it's bad for you. It was a calculated risk. And so when I would do measurements of the residual cyanide, even in the cooked product, there was still quite a bit. But what really got my goat was driving back from Monrovia to the small town where we were living in Liberia at the time, and seeing a woman walk up onto the highway peeling a cassava root and eating it raw. And I thought, my God, what is she doing? She was eating it raw. So the idea that people would consume this root without processing it, other than peeling it, was just amazing to me. You can also eat the leaves. These are the leaves, not to be confused with marijuana leaves. But these leaves also contain the cyanogenic glycosides. Now, this is a part of a family of glycosides, and you're probably familiar with the glycoside amygdalin from almonds or apple seeds, or, you know, they always tell you don't eat the cherry seeds. Eat the cherry, but leave the seeds alone. Spit out the cherry, spit out the apple seeds. And cyanide is ubiquitous in nature. It's so common in plants that we feel that there's probably multiple evolutionary benefits for having cyanide. And this is the cyanogenic glycoside. And as Mark alluded to, You have the enzyme on one side of the cell wall, the substrate on the other side. When you pull the root out of the ground, or when you peel it, or when you macerate the leaves, then you put the enzyme and the substrate both in contact with each other. Voila, cyanide. In fact, in the markets in Liberia, they'll take the leaves, pass it through a meat grinder, and you can actually smell the cyanide as it comes up, as the leaves are crushed. So this is what it looks like. It's pan-tropical, initially in South America. Then the Portuguese pick it up in the 1600s and bring it to Africa. And the people do what they always do with roots: peel it and boil it. And it's a Native American cultivar. I think that it was probably initially cultivated about 6,000 or 7,000 years ago. When we talk about coevolution, what we're talking about is reciprocal genetic influences by non-overlapping gene pools. So for example, we eat a tomato. The tomato and us, we don't share the same gene pool. Maybe metaphorically. But in actuality, there's the tomato genome, and then there's the human genome. But the products of the tomato can influence the functioning of our genomes, and our actions can influence the functioning of the tomato genome. So that's what we call coevolution, where the evolution of two non-overlapping organisms, one influencing the other, they will merge together. They will mutually influence each other. And usually we talk about these coevolutionary dyads, that is, where two different organisms come together. But something's really interesting about that, because when you have a successful dyad, it usually attracts a third species. Kind of like how attractive married men are to single women. I don't know, maybe it's something that married men give off, and then single women say, well, the joke is that a guy shows up with a really attractive woman, and the men say, I want a woman like that. A woman shows up with a really attractive guy, and the women say, I want that guy. So there's this tendency for these coevolutionary dyads to attract a third species. And that's what I think was going on in Liberia. And I've tried to very crudely show this here in this diagram. That is, that the diet that the people consume has an adverse impact on both the sickle cell. Well, it's not adverse. It's actually a positive impact on the sickle cell by preventing sickling. But that same carbamylation has an adverse impact on the on the malarial parasite, because proteins of the malarial parasite will also become carbamylated and be less functional. And then, of course, the sickle cell, if it's not totally sickle cell anemia, but sickle cell trait, then it's beneficial in terms of protecting against malaria. And there's another enzyme, glucose 6-phosphate dehydrogenase, which is just a minor enzyme in the hexose monophosphate shunt. It says as you're driving down the glycolytic pathway, you turn left, and there's the hexose monophosphate shunt. And that G6PD helps to protect the integrity of the cell membrane, the red blood cell membrane. So it looks as though the diet can also decrease the active form of this enzyme, which also seems to protect against malaria. So this is why we have this pyramid-like triangular figure here, where the diet is influencing not only the human and the metabolic processes of the human, but also influencing the parasite, in this case, Plasmodium falciparum, that is interested in consuming the same metabolic products from the human. Because the parasite wants the contents of our red blood cell for its own growth and development. But now we're altering their access to our red blood cells through the diet. That's simply human, plant, parasite coevolution, where the evolutionary trajectories of all three species are being modified. They're modifying each other. But it's like a symphony all coming together. So on top of that now, we have the epigenomics. These are the chemicals that surround the genome, and these chemicals can influence gene expression. They don't change the gene, but they change gene expression. And it's so fascinating because it's inherited. That is, you are carrying the epigenetic markers of your grandparents, which is kind of scary. It means that what they were exposed to has an influence on how your genes express themselves. Just before the talk, I was speaking with one of our colleagues here about this recent article that came out. I believe it was published in PLOS, but it was reported through Sigma Xi, that fear has been shown in mice to be transmitted intergenerationally. Fear, mice. Are you kidding me? This is just amazing. So what else? And it looks as though our susceptibility to diseases transmitted intergenerationally. These are not genetic changes, these are changes in the chemical tags that in proximity to the genes can influence gene expression patterns. What a great source of chemical tags dietary phytochemicals are. So what we eat can influence the tags that influence gene expression patterns. So even when we don't see changes in the genome, we can see changes in gene expression. And gene expression is the foundation for the phenotype. And the phenotype is the direct object of natural selection and evolution. Sometimes we've become so genocentric that we think everything is reducible to the gene, but the epigenome, I think you'll show, we'll see over the next decade, is increasingly important in explaining differences, the differences that we see among an otherwise fairly homogeneous species, our species, because we're young and we're evolutionarily young, and we have not diverged from each other that long. I mean, all of the differences that we make such a big deal about are so superficial, almost to be laughed at. So that when our extraterrestrial colleagues come from outer space, they'll have a difficult time differentiating us from chimpanzees, much less into all of our little social categories that we have, So that gets us to this question of coadaptation and, how deep are the links between the biology of plants and the biology of people. And I would posit that the links are exceedingly deep, because as we have modified the biology and behavior of the plants that we consume, they have modified us. They call to us with their chemicals, or they push us away with their chemicals. They distinguish between friend and foe, insider and outsider. How many of you have been to Jamaica and had that ackee fruit? The ackee fruit, if you don't consume it at the right time, it will kill you. It will put you in a coma. What a great way to distinguish between native and non-natives. Apparently we have a difficult time doing that. Wasn't there a passage, and there's probably some biblical scholars here, wasn't there a passage somewhere in the Bible where the Israelites were fighting people and they had captured some folks and they couldn't determine if they were if they were Israelites or if they were the enemy, whoever the enemy was at that time. And so they had them say a word, and if they could say the word, yeah, you know that story? Yeah, so that means that we don't have such remarkable cues as to who's in and who's out, especially, you know, under adverse conditions like war. But plants and response to plants kind of gives us that insight. It's amazing. So our behavior, our biology, our metabolism is very much skewed by the foods that we eat. And because we continue to manipulate these foods like corn and rice and wheat, including now the intentional genetic modification of those plants, we can expect that we're going to see some differences. We're going to see some changes, especially when all of those modifications in the plant abut against human biodiversity. In fact, it's kind of an unknown process. How will people respond to genetically modified corn? Now, we've always been modifying corn. I mean, corn today is more like a candy bar than it is a grain. It's so full of sugars and so little protein compared to the ancestral corn. But all of this is changing us, and I know we talk about it as being a brave new world, and in a sense it is, but it's a brave new world. Much of it is of our own making and our own responses to the manipulation of other life forms on this planet. So I want to end with that and open it up for some questions. So I'm sure I've provoked quite a few ideas, I hope, and let's see what kind of feedback I can get from you now. Thank you all.